Hello. So let's go for the next speaker. He's Professor Dampier. He's going to give us a talk about harnessing RNA nanomedicines for therapeutic intervention in hematological malignancies. Thank you very much.
Thank you.
I'll let you know when, well, you have that.
It's already running.
It's already running.
Okay, thank you. I want to thank the organizers for the invitation. And I was asked this morning to change my slide and start with a bit of talking about innovation, which is not really related to my lab. I hope so. Well, anyhow, we're trying to combine, think about innovation as a very difficult issue because we have to basically, get out of the box. And I don't like all those cliches, but I'm going to show you, and if people here knows this, so be nice to the others, give them the opportunity, but I'm going to show you that we are all thinking in the box, not outside the box. And maybe this is the reason that we have some issues. So, innovation is the cumulative effect of creativity, motivation, persistence, a lot of luck, and hard work, which is trivial, right? How do I make it? Okay. So how do we solve problems? Let's imagine that the car has fallen into the river. And what will be your ultimate goal to get the car out of the river? How are you going to do this? Yes, okay. Be active. I'm going to show you a picture. Okay, so this is a classical issue. A car is falling into the river, and the majority of people think alike, and they will bring a bigger car to take the small car out of the river. However, only sharp people or physicists, only sharp people or physicists will really look at this as a physical problem because, look at this, the car is actually full of water. Now, the mass of the car is not as the one that was calculated, and the momentum is different. So the next step that will happen is this. Okay, now we have a bigger problem because we have two cars in the river and not a single one. But we are great thinkers, and we now have a better option to cure this incident. And how we cure it? Bring a bigger car, right? So now the big car, which did not calculate the fact that these two have been in the water and are full of water, the mass is very, very big, the momentum will not work, and of course, that will be the unfortunate situation. So this is the thing. We are thinking in cycles. Very trivial, very known, happens a lot. And we need to break this cycle. And in order to move this into my lab, I have to really change the topic here. What's going on? I have to change the topic. And basically going to tell you how we're trying to solve some very simple problem, not curing the world. So our lab is studying how to manipulate leukocytes' function, or basically cellular function, in order to create new approaches to treat mostly leukocytes-implicated disease, because we believe that everything is related to the immune system. When we have this statement 10 years ago, people thought we were crazy, but now apparently it's been very active in different fields. And we are active in hematopoietic field, inflammation, viral infection, and some solid tumors, and some rare genetic disease. All of them has basically one common issue, that leukocytes are involved, subsets of leukocytes. And the lab is divided into two major groups, people that are doing drug delivery system, basically lipid-based and sugar-based, but also lots of protein engineering and antibody formation. And of course, the other side is doing kind of biology, exome sequencing, transcriptome analysis, in order to find new targets and to combine those two arms into personalized approach. Now, we are interested in leukocytes biology. And this is really a critical issue because, over the years we had the pleasure of doing knockout, knock-in mice, etc. to study. But from the fact that nucleic acid, more specifically, the discovery by Craig Mello and Andy Fire about this phenomenon that is calling small interference, or basically interfering RNA, and then the work of Tom Tischel that have shown that it can actually work in mammalian cells. For us, it became a great opportunity to study leukocytes biology in a very short time because we actually manipulate leukocytes function in vivo. And what I'm going to show you, before I'm going to have a few statements, what I'm going to show you is how challenging it really is, but I have also a positive message at the end. So basically, delivery of nucleic acid and mainly RNA, it could be an sRNA or messenger RNA or other parts of RNA, there are a few requirements, because look at this. These are lymphocytes illustration, and this is about 85 to 90% of the surface, or of the mass, sorry, it's a nucleus. And if it's an activated lymphocyte, it will be like 75 to 80%. So we don't have a lot of space in terms of cytoplasm to really do RNAi work here, right? So it has to be really precise. And of course, there are all the other challenges: efficient RNA encapsulation, specificity, which is true for everything, evading clearance mechanism, and avoiding toxicity and immune activation, and of course, internalization, because we have to do it very quickly, and of course, avoiding or try to avoid endosomal, or hopefully creating an endosomal release effect, which I think it's less problematic in those cells. I want to talk about one disease, because for us it's a proof of concept, and this is mantle cell lymphoma. This is the work of 2 graduate students in my lab. Mantle cell is a very aggressive B-cell malignancy. It's have a very low incident. The rate men to women is 3 to 1. It has a median age of about 60, and overall survival is about 5 to 7 years. However, and this is an example of a person, it's a PET scan of a person that has mantle cell. As you can see, it's a bone marrow disease. It actually can spread around very nicely to lymph nodes and other area. So this disease has a very unique genetic profile, which we really like. And in 85% of the cases, or actually if I look at reservoirs and information like the atlas, it's about 83%. In 83% of the patient, there is a chromosomal translocation that happens where the gene that encodes cyclin D1, CCND1, juxtaposed into the Ig heavy chain promoter, which now produce a lot of cyclin D1 in B cells, in normal B cells, and they become malignant B cells because they are now proliferating. So if I now take each one of you and probe for cyclin D1, in normal B cells, the level will be either not existing or a little bit. But then, if we activate the cells, that will jump dramatically, and you can see this happens in mantle cell, and they will start producing a lot of this protein, and basically the cell cycle will get crazy, and at the end, we'll have highly malignant cells that are B cells that proliferate like crazy. And what are the therapeutic avenues? Right now is chemotherapy cocktails, rituximab, and new therapy, which is proteasome inhibitors and ibrutinib, which was approved actually a year and a half ago. For mantle cell lymphoma, it's a B-cell receptor inhibitor pathway. And it looks very promising because the survival has been extended. However, this is only the first event. Then there are lots of other secondary chromosomal changes that happens, and it's a problem. So over the past few years, we work with a company named Alnylam, which is the leading company for nucleic or RNAi approaches, and we have screened with them 510 sequences that can target both the long and the short transcript. And I want to say that most people either have this one, that one, or combination. So we wanted an area that really will cover both of them. And we did a lot of bioinformatics and chemistry and electroporation and everything you want. And at the end, we found out a sequence that actually can work in a picomolar concentration. So this is a very potent therapeutic sequence that actually also create an apoptosis at the end. It's blocking the cell cycle in the G0, G1 phase and create apoptosis. But then, we don't want to electrify people. So the challenges, of course, for siRNA delivery is known. You need to protect them. You need to direct them towards tumor cells, cytoplasm. B cells are really notoriously resistant to transfection. Even viruses have a real issue to transduce B cells, primary B cells. About 5 to 10% will be really great. And, of course, we have the million-dollar question does efficient transfection generate a therapeutic phenotype? So if we see cyclin D1 is going down, does it correlate to really a protein phenotype? It means not only that the protein level will be down, but also that we really see therapeutic effect. So we used the microfluidic system, and we created, that actually I'm doing some publicity here for the precision guys, okay? But it's actually a great system. We synthesized a classical ionizable lipids in this work and in those few works as well. And we created a very robust packaging using the ionizable lipids. And we have shown that actually we can silence in a cell line nicely, about 50%, a little bit more, this gene. And also it blocks the cell cycle. But is it enough? Of course, the answer is not. So we wanted to see if we can create a targeting approach. And we have been working in targeting since 2005. And in 2008 and 2007, we published a few papers, including Science and PNAS and others, that were shown that it's actually doable to do systemic delivery of nucleic acid into leukocytes and subsets of leukocytes. And one of the things we wanted to see if we can actually increase the concentration, the local concentration in the tumor cells, and reduce the off-targets effects. Now, how do you choose the receptor targets? Because we want to direct it specifically to those cells. So we define this as looking at highly or differentially expressed on target cells, and that binding shall be followed by efficient internalization. And of course, there is also all the toxicity issue, immune activation, cytokine induction, interferon response, etc. So you have to make sure that your formulation is not provoking a robust immune response. And this is the thing that we have looked. So for example, these are 4 classical cell lines for mantle cell lymphoma, and we look at different receptors, CD22, LFA1, alpha-L beta-2 integrin, and CD38. And you can see that some of them have different expression level, but CD38 was very promising, but at least in the cell line it looks very promising. So we decided to use this, and we have an antibody against CD38, and we created this production strategy using the microfluidics with the lipid that we synthesize, we mix them, we package them, and the packaging is very, very good and robust. We dialyze it, and then we reduce the antibody, and we can get attachment to the surface, but then we have to purify this. It's a very, very long process, and at the end you can collect those particles and validate. So really, if you want to get rid of graduate students, this is the way to go, right? So those 2 guys that you've seen in the beginning have produced 1 liter of those particles. Why? Because our collaborators wanted to do the experiments blinded. So we shipped them blinded, lots of particles over the years. Okay, so this is a really nice thing. And of course, when they graduate, they say, we are going to become clinicians. We don't want to work in the lab. Okay, but we actually solved this problem as well, and we'll have a paper coming soon also on this thing. So just to show you some data that was published very recently, an anti-CD38 monoclonal antibody, this is the expression level in different cell lines. This is a model I'm going to talk a little bit later, but actually it reached the bone marrow, and you can actually sort those cells, and you can see that the antibody itself internalizes. Okay? The antibody is now in a phase 2 clinical trial. It was in New England recently. It's a very successful antibody. 2 companies have the same targets. And it looks, at least to us, very promising, and also to the New England people. So we reduce this antibody and immobilize it on the surface and clean it, etc. And then we do a co-culture system. We use this. We created a GFP version of those mantle cell lymphoma cell line that have these CD38 receptors. And we took a cell line that lacked the CD38 receptors, and we did a co-culture system. We incubate with a particle that inside have a this is Alexa 647 siRNAs, and you can see that it binds to the granta in the co-culture and not to the TK1 cell that's lacking the expression of the receptor. And then we incubate it with an isotype control, an antibody on the surface that do not recognize these particles. And we also did a blocking experiment. We block with anti-CD38 monoclonal and came up with those particles, and you will see that the blocking is really effective. We also did a shear flow condition where you see a non-coated, they are not penetrating into those cells. and isotype control particles, and the anti-CD38 that penetrate very nicely. And you can see that the rest is a nucleus. So it's not really a lot of work, a lot of space to work. Okay, what about silencing in vitro? So cyclin D1 has been reduced, and we have done all the control, relevant control. Then we move to primary samples from the hospitals. We don't have a lot, but this is just an example. You can see here different patients that have different profile, but they're all CD19 positive, CD5 positive. And CD5 for non-clinician is a T-cell marker, but in this disease and other B-cell hematological malignancies also appearing. And we have done exactly the same experiment, look at CD19 positive and negative with this siRNA inside, and we have seen it binds to this population. With the isotype control, it's reduced, and with a blocking strategy, it's dramatically reduced. Also, it can silence the protein level. But the million-dollar question was, what can we do in vivo, you know? And we found that there is no mantle cell lymphoma model. Unfortunately, we didn't know that. So we had to develop a model. And in order to do this, the same poor graduate students worked for 2 years calibrating a model. That means that they use a GFP version, injected IV, and basically looked at signs of either, you know, IVIS or other style of histology, or just fax analysis in different days, and until, you know, there is some fenestration, some changes in the body weight and manifestation of the disease. And basically, we've calibrated this model, and at the end, we use a SCID CB17 background that we can inject IV 2 million cells, and they basically created this disease. It's a bone marrow disease, I'm going to show you, and 24 to 30 days later, they will die. And basically, they accumulate mainly in the bone marrow, but then here you have an example of GFP and human CD45, which is a marker of leukocytes. So you can see a little bit in the liver, lung, spleen, but the majority actually goes to the bone marrow. And this is another example. We actually can do PCR on this. This is a normal bone. This is a tumor that grows inside the bone. Okay, we can sort the cells, and this is a sorted cell. We can do a PCR, and you can see that the bone marrow really flag here. So we also did this experiment. Again, this was in collaboration with Alnylam, basically a binding experiment. And you can see that the human cells uptake very nice, these guys, and the isotype control much less. But the murine cells also uptake it to a lesser extent and non-significant between those two. And that is because we have a complete FC portion of an antibody. Okay? And again, that means we have to develop a strategy either to cut the FC and use Fab2, or other strategies that hopefully will come in publication very soon that we have done, basically an insertion option. And this experiment, again, was a therapeutic experiment that was done blinded in Charles River. And what you can see here is basically IV injections. It's a retroorbital injections. You can see two things, actually three. One, that this anti-CD38 antibody works. The problem is that a single siRNA, therapeutically or not, is not enough to cure the disease. It's a very aggressive disease, and they will all die. It's extending the survival. It's a dramatic extension in this disease, and it's a very robust model. But it's not enough. So what we are doing now is combination studies with different pathways. As remember, this is the first siRNA, basically, or the first model in this work that was done. But this is a platform. So actually, you can put different antibodies on the surface. And this is a paper that will come this week in Science Translational when basically we look at the strategy to put any different type of antibody, and this is against selectins. So my take-home message is very, very simple. First, the use of microfluidics mixers makes our life a bit easier. You will always need the lipids, but this is a different story. But it makes a robust production, easy to control, and scalable. And then the novel delivery system are being developed in order to really achieve this active cellular targeting. And of course, it's all in the message. And I want to finish by saying really thanks to the people and the people that support our work. And mainly this was supported by NCI, the National Cancer Institute, Alnylam, the Israeli Science Foundation, and the Broad Institute that help us with the bioinformatics. And thank you all for listening.
Thank you very much. We have time for questions. Please.
Thank you very much for your talk. Congratulations to you and your two students. You've done a great job.
They're not anymore with me.
Regarding the last slide that you had with the therapeutic effect of the nanoparticles with the antibody and with the siRNA, I have two questions. The first is, you show there that with control siRNA, in the nanoparticle with the antibody, you have some effect there.
It's
Is
the
that
antibody.
related to the presence of the antibody?
Yes.
And in that case, do you know if you were only administering the antibody without the presence of the nanoparticle, will the effect will be the same in that case?
Less.
Less?
Yeah.
So
we
that's
have this control.
That's
It's a very
something
important
that you should
control.
explore in then, though?
Yes. It doesn't, so if you don't have an antibody on the surface, you have a problem. It will reach, to some extent, the bone marrow, but we have very low amounts, 1 or 2%, if you quantify this. And then also, I don't know if you noticed, but we have injected, Charles River injected a lot of time. Yeah. This is a very aggressive model. Okay, so the model, those mice die very quickly, and they're proliferating very quickly, much more probably than the human disease. But it's a very good model because, for example, ibrutinib, that is a difference, we are collaborating with another company. Ibrutinib works very well in this model. But they will all die before the antibody. Okay. So still, I would say it's not a translation, it's a research thing, but yes, it's a very important control. It was included originally.
Thank you.
Please.
So were you able to assess the level of knockdown of cyclin D in vivo?
So we have not looked at the level of knockdown in vivo, but basically we can sort the cells, and the assessment that we have in a different siRNA, so I don't know about cyclin D1, but for PLK1 I have data that will be published also very soon, that we have reached about 75% knockdown. And we block G2M there. Okay. So it's, to some extent, even more robust than cyclin D1.
Okay, so thank you very much again for your
Thank you.
intervention.